for being here. I'm just going to give you a little overview of our program and what we do and what we offer and what we can offer both to you, but more importantly, what we can offer to the patients of the community and beyond. So I spent 3 weeks, a couple weeks ago, I just got back from Africa, and I learned a lot about a lot of things. You know, I always used to say that my feral colony in the backyard taught me 3 things, which are very, very pivotal and relevant to cancer drug development. They taught me patience, perseverance, and persistence. And I can tell you that after camping in a tent without internet or cell phone access for 3 weeks in the Serengeti-Gorgora corridor in Tangier, I learned that these cats probably have taught me more than my feral colony in my backyard that I've cared for 8 years. Amazing animals, but I think the most important thing was spending a day and a half living in a Maasai village, which actually changed my life and actually changed my trajectory on where we really need to go with clinical research. We need to go to the last untapped frontier, which is Africa, which we are currently working on a project to be able to bring clinical trials there, thanks in large part to many of the companies that are currently in this room today. This is my phase 1 team of clinicians. The people on the top are the physicians that are on the team, and on the bottom are my advanced practice providers. And as you notice, they're a little bigger than my physician pictures. The physicians are extremely important, but the backbone of my team, and I have to thank them every day, are the APPs that make it possible. And many of you have worked with them in the past. And this is my CTO phase 1 team. We started with 3 people when I got here 11 years ago, and we've built it up to over 45, 50 people. I'm very, very fortunate to have such a dedicated team because I know that there are a lot of other jobs that they could go to, and they stay with phase 1 because they believe in the cause. So one of the things I want to bring out is the hybrid decentralization program. This was an idea that I came up with a few years ago because we found that we were not getting a large number of patient referrals from certain communities in the area. And when I looked at it, it was the underrepresented communities. And so I wrote a series of grants, and I was very fortunate to get them funded, some by peer review, others by pharmaceutical sponsors that have helped me. And for those of you in the audience, again, thank you. But what we realized is that a large number of these patients were not being referred, and they're actually patients that too could benefit from our program. And so we wrote a grant based on 3 aims. The first was to figure out from both the patients as well as their caregivers, and especially the providers who would otherwise refer patients to us, what the problems were. So once we identified that, we started tackling it to implement going into the communities to be able to provide early phase clinical trials to these patients in various approaches. So the New Haven campus initially expanded to Bridgeport Trumbull, and I thank all of the physicians there who have been very instrumental in referring patients to us. It not only increased the number of patients we put on trial, but increased the number of underrepresented populations, included the underrepresented that are the rural poor or the urban poor. And starting in October, the team will be moving also to opening up a clinic in Waterbury, Torrington, to make it more accessible for the patients that otherwise would not be able to come to us. Just a brief overview, this is our current portfolio. It involves 62 phase 1 trials. However, not all of these trials have openings as we want them because more and more pharma is increasing the number of sites to early phase clinical trials. And so we're always bidding for additional slots. And that's why you may see that we have 14 different ADCs, or we have 4 KRAS G12Cs or Ds, because we try to increase the portfolio as the patient referrals come in so that we're not necessarily on a stop and go at all times, but we'll be able to access trials slots for our patients as they come through. And we also have many trials in startup. As you can see here, just looking at the antibody drug conjugates that we have several antibody drug conjugates that are currently open. I believe there's 17 of them soon to be 17. And many of them, as you can see, have multiple different targets. The problem we're still confronting is that the payloads are very similar. So as you can see, the majority are topo-1 analogs based off the parent topo-1 inhibitors. And we do have a couple MMAE inhibitors, which actually are coming back into vogue only because of the fact that topo-1, there's about 168 active ADCs that have topo-1 payloads that are in clinical development currently. But what we are also doing is working closely with pharma to get very, very unique payloads coming through that I consider high risk, but also high reward. And you know, many of these novel drugs that we have in our portfolio, sorry about that, are actually, even though KRAS G12C inhibitors, there's two of them that are FDA approved, sotorasib and adagrasib. Next generation agents, the next generation G12Cs, have significantly, although there's not been a head-to-head randomized phase 3, and there will never be, have significantly greater efficacy and durability in terms of response than the current FDA-approved ones. So if in doubt, and you have if you have a patient with a G12C alteration that has significant allelic alteration that has not yet been treated, we urge you to contact us so that we can hopefully give them the next generation agent. So just looking a little bit at our program. So obviously, almost all the phase 1 trials that come to the main campus are done through the early phase clinical trials disease team. We do that for many reasons. You know, I've been doing this for close to 40 years, so I understand the reason of having your team in decent control to minimize deviations, violations, which unfortunately do happen. I see them especially at other sites. And there's something to be said about experience doing these trials. When they go into phase 1 expansion, as you can see here, they'll either stay on the early phase team if they're basket type trials, or they'll go to disease team, specific tumor disease teams. If they go on to, if these drugs make it in phase 1 and go on to phase 2, 3, we really push hard on our team to discuss with the sponsors to bring those trials to Yale and give them to the respective tumor disease teams. I'm going to talk to you specifically only about 2 of them, but as another example, being the president of AACR, I was able to be extremely involved in the development of a neoadjuvant trial for gastroesophageal cancer. And I know that the GI team, I think, is going to be one of 5 sites that AACR has chosen where we're going to give novel therapies, some of which have not yet been FDA approved or FDA approved for their indications in a neoadjuvant setting. Because as you've heard from Raghav, GE cancer is a horrible disease with significant morbidity and mortality, unfortunately. So if we can bring those drugs earlier into the neoadjuvant setting, hopefully we'll be able to, number one, our goal is to prevent gastroesophageal resection so that we can minimize the morbidity of these patients and hopefully elicit a greater degree of cure. Many of those drugs that are going into the AACR neoadjuvant trial were drugs that we've worked on and that I've had extensive experience with. So that's an example of a neoadjuvant trial that's going to be coming forward. But I want to talk to you also demonstrating this pattern of how phase 1 feeds into other tumor types. I'm going to talk about these 2 drugs in particular, the KAT6 inhibitor, which is profetrostat, and the c-MET inhibitor, which is TMAB-A. Let's just start with profetrostat. This drug is a KAT6A/B inhibitor. It's an epigenetic targeted drug. Previously, for many, many years, we've worked on epigenetic drugs in the solid tumor arena, and unfortunately, they've been pretty negative. However, recently, with the EZH2 data, we've seen some real promise with epigenetic drugs, and with the KAT6A/B inhibitor, I hope you'll see or agree with me at the end of this that it is also a very exciting drug. Not only is KAT6A/B inhibitors exciting, there's more than one. This is one that, as you'll see here, has been developed by Pfizer. There's many other companies now that are developing these. Menarini Stemline has one. We've got the phase 1 trial of that open as well. And there's KAT6A selective because many feel that the dysgeusia could potentially be from the B inhibition, although I'm skeptical of that. And so there's other drugs as well as CAT2, CAT7, et cetera, coming down the pipeline for various indications in various solid tumors. So this was a study that I presented at ASCO 2004 as an oral, and we were able to update it with, believe it or not, another oral at ASCO in 2005, where we developed this drug in dose escalation and then moved forward to dose expansion. And thanks to a lot of you in the audience at the surrounding centers, as well as in other practices as well, we were the lead recruiters putting approximately 24 patients on this trial. So KAT6A inhibition represses estrogen receptor transcription and hence also overcomes ESR1 mutants that confer resistance to endocrine therapy. So orthogonal inhibition of other oncogenic pathways driving hormone receptor positive breast cancer are also affected, as you can see, by KAT6. This drug preclinically demonstrated suppression of estrogen receptor in an ER-positive tumor model, as you can see, vehicle versus oral drug in the mouse, and in several preclinical models, but as you can see in this one, in this PDX hormone receptor positive model, we got significant inhibition with the KAT6 inhibitor, 3.3 milligrams, 1 log different, 3 milligrams relative to the vehicle control. So we embarked on the phase 1 trial, and based on preclinical data, we began at an 8 milligram dose. And then we escalated to, as you can see, 15 milligrams, subsequently got the pharmacokinetics of the first cohort, realized we were over significantly in terms of exposure, dropped the dose down to 5, dropped it down to 2, dropped it down to 1. So this was an escalation followed by multiple de-escalation steps. Then what we did in part 1B is we tested it in combination with fulvestrant to determine whether or not there would be any enhanced toxicity of either the fulvestrant or the KAT6 agent. And this was our phase 1 dose escalation for safety and toxicity. As you can see, based on linear kinetics, it was very, very linear. This is exactly what you want to see. Very good performance of a drug in terms of PK. And when we looked at PBMCs to look at pharmacodynamic endpoints, looking at H3K23 acetyltransferase, we were able to elicit at all cohorts significant decrease. We were definitely hitting the target, at least in the peripheral blood. And we did serial biopsies on a select group of patients, and between baseline biopsy and day 15, we were again hitting the target. And you'll see that we only looked at it at 1 and 5 milligrams because those were the 2 doses that we were going to advance forward based on Project Optimus to identify a potential dose to advance into phase 3. After the phase 1 dose escalation, we then identified, as I said, that it was safe to give in combination with fulvestrant. And so what we wanted to do in the second part of this study was to determine whether or not there was anti-tumor activity of monotherapy. These patients were second line and above. Some patients had as many as 8 prior treatment regimens. And we wanted to see what it looked like in terms of efficacy as well as toxicity in a more expanded cohort in combination with fulvestrant. As you can see, 2A looked at the dose that we had selected potentially to advance forward into phase 2/3 trials, which was 5 milligrams. This was the monotherapy at 5 milligrams. There was efficacy there, but it wasn't earth-shattering. But if we looked at the overall response rate, it was only 11%. But the duration of response, when we finally analyzed the data, this was the data that was originally presented, was about 15.6 months median. And this was with monotherapy, keeping in mind that these patients had a median of 4 prior treatments, including not only hormonal, but also chemotherapy. The clinical benefit rate, in addition to that, was 31%. And so then looking at it in combination with fulvestrant, same patient population, median follow-up at this time was only 9.2 months. Overall response rate, as you can see, 30%, with a beautiful duration of response. Again, it ended up being 15.6 months. But in these little green triangles, these were patients that had clearly progressed previously on monotherapy fulvestrant. And 3 of them not only had a confirmed PR, but that PR was very durable. And actually, one of those patients is still on study about 3 years later. This is looking at the tumor size from baseline at that recommended phase 2 dose with fulvestrant. As you can see, these spider plots are very, very beautiful. And if we looked at the 1 milligram monotherapy versus 5 milligram monotherapy versus 5 milligram plus fulvestrant, again, a heavily pretreated population, you see that the PFS differential for the combination was sufficient to want to take this combination forward. And as you can see, the overall response differential increases between 5 milligram monotherapy, 1 milligram monotherapy, and the combination, with the median duration of response again being 15.8 months. ctDNA showed significant positive effects, not only overall in the entire population, but also in patients that had known ESR mutant variants at baseline that decreased significantly at cycle 2, day 1, when the second set was taken. The one main problem, it was very well tolerated. We do get dysgeusia, but I can tell you that dysgeusia is a phenomenon that we've seen with other epigenetic drugs, as well as for those of you that treat metastatic basal cell. The hedgehog inhibitor, when we were doing the phase 1 trial of that, also caused dysgeusia. Some of the other epigenetic drugs are also causing dysgeusia. We're working diligently to try to overcome that toxicity. However, as I said many times, we take phase 1 drugs that come through our team and we push them into the disease teams when they're advancing. This trial, I'm told, will be opening up hopefully within the next month. The PI of this trial at Yale will be Adriana Kahn, who also does phase 1 trials with us, as well as breast. And it's called the CAD-CIS trial. I thought it was really kind of cute. They paid over $100,000, by the way, to name this trial. I found out from the medical affairs people. But this is the trial, and I think it's going to be exciting. And these are the inclusion-exclusion criteria, and hopefully many of you at the outlying centers that are part of the Yale system will open this up at your sites as well. This is a trial and a drug combination that I think is quite exciting. We had, as I said, 2 oral presentations at ASCO. There was an oral presentation by one of the investigators at San Antonio 2024, and I got notification from San Antonio yesterday that this is going to be accepted in San Antonio 2025 as well. We got a Nature Medicine paper that came out, ASCO 2024, the same week that the data was presented, and we have a dose optimization manuscript in preparation. We should be submitting it to JCO soon, just to show that many of these trials not only lead to other drugs, but they also lead to, I think, very pivotal publications that I think are a credit to Yale and what the Yale Cancer Center is doing in terms of supporting our program. We just recently, about 2 weeks ago, got this LOI accepted. They're not necessarily looking at ESR1 mutants, as you could see from the phase 3 trial design. We think not only is it important to understand, because CAD6 expression does not necessarily predict for efficacy. So we submitted this LOI and got it accepted less than 2 weeks ago. So we're starting to move forward with the clinical trial to better understand this mechanism of response, mechanism of resistance, because as I showed you, it's very encouraging that roughly 40% of patients have a response and up to 60% of patients have clinical benefit in a heavily pretreated population. But the question is why? Because with other drugs coming down the pipeline that may be as exciting, we want to be able to pre-select our patients that we feel would best benefit from even these phase 3 trials. So we're pretty excited to advance this concept forward into a clinical trial that's being developed. The next drug I want to talk about is TMAB-A. This is the ABBV-400 drug. This is a c-Met antibody drug conjugate. I think it has a DAR of about 6. Its payload is a topoisomerase inhibitor. We were involved in this phase 1 trial. Dr. Mike Cicchini, who does GI in phase 1 and who just gave an excellent talk about an hour before me, was pivotally involved. He was a site PI for this trial. The dose escalation, as you can see here, was recruited a 3rd line in greater colorectal cancer that were c-Met unselected. And then there was a guided BOIN design. We initially had more than just colorectal cancer recruiting to this trial, and that's why it came onto the phase 1 trial, the phase 1 team. Eventually, though, they were looking specifically at third line and above colorectal cancer. We do know that c-Met is expressed in this tumor, and chemotherapy can enhance the expression of c-Met at the time of as a mechanism of resistance. And then they went into a dose expansion trial with third line and greater colorectal cancer. And throughout the dose escalation and expansion, they did not pre-select for c-Met amplification. But in the phase 1 trial, the primary objective was safety and tolerability. As you can see, this study was presented by Dr. Sharma. We were late to the game in getting it open. So Dr. Sharma and Powderly were the lead and senior investigator. And what they showed in this presentation, these are specifically colorectal cancer patients advanced, that there was activity even with the first dose of ABBV-400. It increased at 2.4 mg per kg, and it was really prevalent. As you can see, 24% response rate at 3 mg per kg with a complete biologic response of 68%. Many of these, the median was 4 prior lines in this recruitment. So this was quite a paradigm shift in what we see with third line treatments such as LONSURF, et cetera. Very, very exciting data. So then what they did is they went back and they looked at the c-Met expression. And in those patients that had a c-Met cutoff of greater than or equal to 10%, there was a 37% response rate. All patients 20%, and those that had a cutoff of less than 10%, 14%. But as you can see, the median PFS with those patients whose tumors were c-Met greater than 10% was 5.4 months. Very different than LONSURF. So just as I'm finalizing here, I just want to show you, and thanks to Dr. Cecchini for building this slide. We were very involved in the first in-human study, although we didn't activate it until quite a bit later. We then started recruiting, and in that initial phase, we were able to put 6 patients on. It eventually then, not only did it expand to multiple tumor types, but subsequently went specifically to third line colorectal. At that point, we transferred the study to the GI team, and enrollment was complete, as you can see there. And we were able to put 5 patients in the expansion cohort. This has led Dr. Cecchini to an oral presentation that will be upcoming at ESMO 2025, as well as another oral presentation for which he will be co-authored on. But I think what's most important is being integrally involved in that phase 1 trial and proving to them that even though we were late in the game of opening, we could still recruit better than any other site that had the trial open. It is allowing us to have open at Yale a first-line study, a second-line study, and an open-label randomized study that Dr. Cecchini is on the steering committee for. This, I think, is a great advancement to GI cancer, specifically colorectal cancer. And I think these trials will be a gift to our patients as well as the oncology community at large, because I have high hopes for this drug. This is how you refer a patient to our team. These are the docs. It's just our first name dot last name at yale.edu, or you can call the clinic to set up an appointment. We encourage you to contact us. We get back to you. We see the patients within a week of being referred to us, if not sooner. And I'd like to close with this male leopard. He was gorgeous. He was amazing. We were able to see him. He's sitting in a cliff as the wildebeests are crossing the river for the Great Migration. He's just picking out which wildebeest he's going to go after. And actually, we saw him do it. It was amazing. And with that, I thank you.
Okay, Stacy. Pat, you can stay for 1 second. So that was great. You know, I don't want to go too long because the wildebeest is eating, but we won't eat if we go too long. But listen, two things. This is phenomenal. You know, the phase 1 group. If you can refer to someone, they're all off presenting abstracts at ESMO and ASCO. But Pat, how does one refer? So let's say someone has a patient with breast cancer, and they're out of options. Do you refer to the breast group at Yale first, or would you rather they refer to the phase 1 group, or is it an iterative process?
Well, it can be an iterative process, but if you contact us, we let you know immediately if we have something, and typically Adriana or I will pick up the breast patients because I've been very involved in breast cancer all my life. And when you email us, we get back to you or text us, like Dr. Frank in the community texts me constantly, and I can immediately get back to him and let him know whether or not we have any trial for the patients. And if not, we'll send them to the breast team. But more and more, these trials, even in phase 1, when we get into expansion, are requiring less and less prior treatments, especially biotech, because they need to get a hit for their VCs and their investors. So we pretty much know what's going on, and that's why each of those junior faculty or an intermediate faculty represent one of the specific DTs. So we have GU, GI, breast, and sarcoma that are representative of the faculty on our team, and lung, Sue Young.
Terrific. We probably have to move on, but thanks.